Hello everyone! Today, I'll be presenting part A of our assignment, which is about tilling, a fascinating plant breeding method. Tilling stands for targeting induced local lesions in genomes. This method is widely used in genetics and agriculture to identify mutations in DNA. For our example, we've focused on tomato mutation breeding, which shows how this technique improves crop traits like yield, pest resistance, and stress tolerance. Induction of mutations. The process starts with treating tomato seeds with EMS, ethyl methane sulfonate. EMS is a chemical mutagen that alters the DNA, introducing random mutations. These treated seeds become the M1 generation. This step is crucial for creating genetic variability, which can result in traits like higher yield or disease resistance. DNA preparation and pooling. DNA is extracted from a large number of M2 plants, about 15,000. The extracted DNA is then divided into smaller pools. In tomato research, this pooling step ensures we can efficiently analyze large populations to pinpoint useful mutations. PCR analysis. Next, PCR, polymerase chain reaction, is used to amplify specific DNA fragments of interest. Multiplex PCR helps amplify multiple regions simultaneously, while simplex PCR is used for detailed analysis with LC green, a dye that detects mutations. For instance, a mutation in a tomato gene might improve shelf life by slowing down the ripening process. Mutation analysis. High-resolution MELT (HRM) analysis is performed to detect DNA variations. HRM helps identify mutations with high precision by studying how DNA strands separate under heat. Once mutations are detected, tools like the ABI 3130XL are used for sequencing and mapping. This allows researchers to confirm and understand mutations that, for example, make tomatoes more resistant to drought. Definition. Mutations are changes in DNA that create genetic variability, essential for improving crop traits. Purpose. Tilling helps identify and utilize these mutations to develop better crops. Materials. For tomatoes, we used EMS treated seeds, PCR machines, HRM with light scanner, and sequencing tools. Diagram. The process is visually summarized step by step for easy understanding. The example of tomato mutation breeding shows how tilling transforms agriculture. Mutations identified through this process have led to tomatoes with higher yields, better taste, improved shelf life, and resistance to pests and diseases. This makes tilling a crucial tool for modern crop improvement. To summarize, the tilling process involves inducing mutations, cultivating plants, extracting DNA, and using advanced molecular techniques to detect beneficial mutations. This method is valuable for creating crops that are more resilient and productive, which is crucial in modern agriculture. On behalf of Group 4, I'd also like to share our poster, which summarizes everything about tilling. It includes the definition, purpose, procedure, and key materials like EMS, tomato seeds, PCR machines, and light scanners. A diagram is also provided to illustrate the steps clearly and concisely. Thank you for listening.